All right, let's get into it. We usually think of myelin as just insulation for our nerves, right? Kind of like the plastic on a wire. But what if it's doing something way more important? Today, we're going to look at a study that completely reframes myelin, not as some passive wrapper, but as the active conductor of our body's entire electrical symphony. I mean, just stop and think about that for a second. Your nervous system is this incredible biological supercomputer. It's processing information at speeds that are just mind-boggling, letting you think and move and react in the blink of an eye. The complexity is just wow. So inside the supercomputer, the signals are zipping along these biological wires called axons. And to make them ridiculously fast and reliable, a lot of these axons are wrapped up in that fatty stuff called myelin. And that's really where our story kicks off. Now, for years, the textbook definition of myelin has been super simple. It's insulation. Its job is to stop the electrical signal from leaking out, which helps it travel faster. Simple as that. But as it turns out, that's not even close to the whole picture.
So back in 2016, a study in the Journal of Physiology asked a really cool question. What if myelin is more like a musical conductor? What if it's actually organizing all the little parts needed to make sure that nerve signal plays its tune perfectly, especially right at that critical moment before it gets to its destination?
To figure this out, scientists zeroed in on one very specific, very important spot, the axon hemonode. You can think of this as the final checkpoint, the last stop for a nerve signal before it makes that leap over to the nerve terminal to talk to the next cell. It's a critical handoff point.
And the way they tested this was honestly brilliant. They compared a normal, healthy rat that has this perfect, compact myelin sheath to a special kind of rat, the Long Evans shaker rat, which has a genetic quirk that means it can't form that compact myelin. So what you get is a perfect side-by-side, a system of perfect order versus a system of total chaos.
Okay, so first, let's look at the healthy system, the well-conducted orchestra. What does this state of order actually look like when you zoom all the way in? In a healthy nerve, the sodium channels, which, let's be honest, are like the first violins of this orchestra, they are lined up perfectly. They're packed into this tight, dense little cluster right at the hemonode, exactly where they need to be to give that signal one last, powerful push. And the result of this beautiful organization is, well, it's perfection. When the signal hits, almost all the sodium floods into the nerve at that one precise spot, the hemonode. The actual nerve terminal itself stays pretty quiet. It's just a clean, powerful, and incredibly efficient handoff. All right, now for the fun part. Let's see what happens when you take the conductor, that compact myelin, out of the picture. This is where things get really messy. So in the rat without proper myelin, it's a completely different story. It is a total mess. Without myelin to organize everything, those sodium channels are scattered all over the place. They're not in a tight bunch anymore, they've wandered off and have even invaded the nerve terminal, a place they have no business being. The first violins are just wandering all over the stage. And the consequence of this chaos is huge. The rush of sodium at the hemonode is way weaker now, and you've got this improper signal leaking into the terminal. The signal isn't clean and focused anymore, it's just smeared out and disorganized. So, as you can probably guess, this structural chaos leads to a pretty catastrophic breakdown in how the nerve actually works, especially when you ask it to do something really fast. So here's the key. There's this really special electrical property called the resurgent sodium current. Think of it like a super fast recharge mechanism for those sodium channels. It lets them reset almost instantly, which is absolutely essential if a nerve is going to fire hundreds of times every second. And just look at the difference here. The healthy rat has this big, powerful resurgent current. It can fire away at high speeds, no problem. But in the dysmyelinated rat, that ability is practically gone. The nerve just can't keep up. The orchestra literally can't play fast music. And the researchers, they actually pinpointed the culprit. It's this tiny but critical protein subunit called NaV-beta4. This is the little guy responsible for creating that fast recharging current. And in the dysmyelinated rat, it was completely missing from the hemonode. Myelin wasn't there to put it in place, so it just wasn't there. And the chaos didn't stop there. Other members of the orchestra, like the potassium channels, were also out of place. This led to all sorts of improper electrical currents. The bottom line? The nerve was less excitable, and crucially, it failed completely if you asked it to fire faster than 300 times a second, something a healthy auditory nerve does without breaking a sweat.
So what does this all mean when you put it together? It means myelin's true role becomes crystal clear.
It's not just insulation, it's the ultimate organizer.
I mean, the contrast here couldn't be more stark, could it? On one side, you've got the healthy state of order. Compact myelin is present, all the channels and proteins are where they should be, and you get a reliable, high-speed signal. On the other side, chaos. No myelin, everything's a mess, and the signal just fails.
And the study's authors, they said it perfectly. They concluded that compact myelin is essential for the orchestration of these ion channels. They didn't use the word insulation, they used the word orchestration. And that one word changes absolutely everything.
So really, the next time you think about myelin, try not to picture just a plastic wrapper. Picture a master conductor, working tirelessly behind the scenes to make sure every single molecular musician is in the right place at the right time so that the incredible symphony of your nervous system can play without a single missed note.
And the precision that this conductor achieves is just astounding. It's what allows certain nerves in your body, like the auditory nerves they studied here, to fire reliably up to 1,000 times per second. And this all leaves us with a really fascinating final thought. If myelin really is an active conductor, that opens up a whole new world of possibilities. In diseases like multiple sclerosis, where myelin is lost, could we find a way to not just replace it, but to actually teach it to rebuild its orchestra, to restore that perfect high-speed signaling? The score is already written. Maybe we just need to help the conductor get back on the stage.